The theory of abiogenesis states that life could have originated from non-living organic molecules present on Earth during the prebiotic time. The first cell-like structures that were possibly made out of these organic molecules are called the protocells. The membrane component of these protocells might have emerged from amphiphilic molecules, which could have spontaneously assembled into vesicles and encapsulated the chemical systems that are required for early evolutionary processes. Protocells, if it needs to be considered as a cell, keeping aside all other cellular aspects or characteristics, should have the ability to grow and divide. There are various proposed methods by which a protocell could grow and eventually divide. It could happen via the addition of fatty acid precursors that are released from either surrounding micelle or other vesicles. And then after the growth of the protocell, spontaneous division could have been occurring in the early times as the only means of protocellular cytokinesis. And this could again happen due to various factors or reasons, like internal forces, which includes osmotic pressure, or mechanical forces, which can lead to purling instability as shown in the figure. To become a modern cell, the protocellular compartments must have made the switch or the transition from having fatty acids as their membrane molecules to phospholipids, as shown as the green molecule. With phospholipid addition to the vesicle, the growth of these mixed lipid vesicles would have increased in comparison to the vesicles which were only having fatty acids. And then, due to the increase of the strength of these membranes with the increase of phospholipids, the spontaneous division have a less chance of occurring, which eventually leads to the formation of new genes and catalysts which would have evolved to make the division process possible. But which catalytic mechanism would have led to the division of the protocell? So there is an idea which proposed that maybe some extant enzyme that are needed for lipid synthesis could have led to the growth and the division of the protocells. However, till date, no such enzyme was found. Hence, a new hypothesis was made which states that maybe the lipid synthesis could have started in an external environment rather than an internal one, and later some ancient enzyme or proteins may have assisted in the division of the cells after it has acquired the necessary lipids. Before going into the catalysis part, I would like to mention the advantages of having a periodically timed division. The cells controlling the timing of cytokinesis by any enzyme or early protein could have gained the advantage of coordinating the replication timing of informational molecule with the early cell membrane budding process in order to successfully and efficiently transfer all of its genetic information and metabolic molecules equally to its progenies. This has led to our hypothesis where we think that FTAZ might be the early protein which is needed for the process of division of the protocell. So what is FTAZ actually? So FTAZ is a GTPase protein encoded by FTAZ gene that assembles into a ring-like structure at the future site of bacterial cell division. FTAZ is found in almost all bacteria, archaea, chloroplast, and mitochondria, that is very much essential for cell division process. FTAZ is also an ancient protein. It contains very less amount of amino acids like arginine, lysine, phenylalanine, etc., which were hypothesized to be the last one to be added to the genetic code. Hence, it suggests that FTAZ evolved as a functional protein way before the genetic code was even complete. And FTAZ is the only gene controlling cellular division present in all resident minimal bacterial genome that are being analyzed till date. The activity of FTAZ on various in vitro and in vivo systems which mimics the protocellular structure has been already observed. Like for example here in the case of unilaminar vesicles, the filaments of FTAZ form patches inside the liposomes, and these patches are reorganized to form ring-like structures which are parallel to the plane of the membrane. Some projections were observed at the outer portion of the vesicle like buds or tubules, and the position of the projections is actually coinciding with that of the rings or the patches that are made by FTAZ, which shows that rings that are made by FTAZ are able to construct the membranes of the vesicles partially, which could be a cue for protocellular division. And the similar things have been observed in other in vitro systems as well, like SLBs or supported lipid bilayers. These model systems, along with FTAC, could be able to resemble the divisional machinery that were present in primitive cells and help us understand the replication pathway. Therefore, our project or our work is focused on the reconstitution of FTAC on various relevant in vitro and in vivo systems like liposomes and others, while observing its dynamics and the ability to divide these protocellular systems. FTAZ usually binds to the membrane not directly but with the help of another protein, which is either an FTAZ-A or ZIP-A, and it binds to these proteins via a C-terminal domain. So before adding the protein on the in vitro systems and doing the studies, we have changed some bits and bits of the protein which could help us visualize the protein better on these systems. So this is a construct, or one can say a plasmid map containing the genes that our design protein is having. So instead of having a ZIP-A or FTAZ-A as a membrane anchorage protein, we are using E. coli Mindy MTS. So FTAZ-A had a profound effect on the dynamics of FTAZ when both of them were reconstituted in phospholipid vesicles, which overshadows its capabilities and functionalities. Hence, it has been replaced by a non-interacting membrane binding protein, which is MTS. And therefore, the C-terminal region of FTAZ is not needed. Hence, the plasmid contains FTAZ without a C-terminal and MTS and a fluorescent tag, which is amnion. In addition to this, we also constructed two other plasmids or constructs that are having GTPase mutant version of FTAZ to see how GTPase mutants can actually affect the protocellular division process. After the construction of the constructs, the protein is harvested and purified from competent strains of E. coli. Because stringent purification processes, the protein is usually checked first before any further downstream processes are being done on it. We perform the sedimentation assay for the purpose of checking the activity. A sedimentation assay is nothing but an in vitro assay to observe the polymerization of the protein. Here in our case, the protein, if active, should be able to form long filaments when GTP is added. And if in the meantime the reaction is centrifuged at high speeds, then filaments should go down in the pellet section, showing us the presence of polymerization. And here one can see that FTAC is being polymerized or being active even after our protein purification process. The amount of pellet in the presence of GTP, as shown in this lane and this lane, is way higher than the control one having no GTP, as shown in the lane 7. And also the concentration of the FTAZ in the pellet increases both with the increase in FTAZ concentration and GTP concentration. The first system that we used is that of Spheroplast. So it is a bacterial cell or plant cell that is bound by its plasma membrane only and without a cell wall. This could stand as a model cellular system which could have evolved during the evolution of early microbes where the cell wall and cell membranes were still evolving. The turgor pressure in this case is very low compared to the membranes in the normal cells, which could be mendable by FTAZ. As it could be easily mendable or deformable, few vasculation events could have been observed in the cells after the addition of the protein. So we took a DH beta cells having our constructs and gave it an induction of arabinose and formed spheroplast and observed pinching at various degrees and in various number as given by the red arrows. This led to the fact that our construct could lead to the potential division of spheroplast or the model cells that we are envisioning. The next system we studied is an in vitro one, the lipid droplets. The droplets were either made up of POPC, POPE, or only POPC. The protein was allowed to polymerize on the top of the droplets. The top row shows the images that are taken in wide field microscopy, where the bottom one were taken with Fitzer filter, showing the protein fluorescence. Binding of a protein was observed in the droplets, with the FTAZ fluorescence coinciding with the membranes of the lipid droplets, as shown by the wide field microscopy. Then we did SIM microscopy and observed the Z section of the droplets and saw that the protein is making some kind of circular-like structure over the droplets. Inside the circles, no fluorescence from FTAZ is at all observed. This points to the fact that maybe the protein might be present on top of the membrane loosely all over the lipid droplets, but in some place they were able to condense to make polymers and could form rings on the droplets, which is a signature move that FTAZ does before constructing a bacterial cell. However, no plausible construction was observed. After observing the proteins on these two systems, we are planning to perform the experiments on liposomes and that are made of mixed fatty acids and see how FTAZ is behaving on them, to see any blebbing or vasculation events that could be occurring. And if it happens, then FTAZ could stand out as a potential candidate for being the primitive protein needed for protocellular division. And the same thing would be observed on SLBs too, to find out about the mechanism. At the end, I would like to thank my funding sources, my institute, NICER, the university under which my institute is, Homi Bhabha, and Department of Atomic Energy, India. The project, the whole work and the ongoing processes will be the part of my undergraduate research project, which is done under Dr. Ramanujam Srinivasan. And I would also like to acknowledge one of his grad students, that's Ajay Kumar Sharma, who has helped me in every process, each and every process of the experiments, and also to the members of Srinivasan and Matthew Lab. So these are my socials. I would be more than happy to take in any questions regarding the work, and will be also happy to share any updates that are happening during the conference. And thank you.